Oh, that's just beautiful. I've been to their campus. It's so cool. Like, the coolest ever. Yeah, and everything's like tucked up into the trees. I first went there and I was driving through the campus and I didn't realize that on campus because everything was like
at the moment it's
where are all the
Okay, let's start the lecture portion. So I mentioned last week we did linear models, and I'm going to tell you now that we did linear models for fixed effects. And I also said that sort of linear models would be a kind of a sketch of many of the methods that we would be encountering subsequently. And so here's the first demonstration of this. I'm going to talk again about linear models, but with a twist. And the twist is that we have data that includes a random effect. And I'm going to tell you what that means and how this works, how we model and analyze data when random effects are present. So first of all, a summary of what the difference is between random and fixed effects. So fixed effects are the things that you want to try to understand when you carry out an experiment or an observational study. They're sort of predetermined categories of a variable, and they're typically of direct interest. And not only that, but they're repeatable in the sense that somebody on the other side of the world could potentially actually redo your experiment and use exactly the same levels of the factor. So examples of fixed effects would include medical treatments in a clinical trial experiment, or doses of a toxin, so they don't have to be categories, they can be continuously varying. Diet or fertilization treatments, age groups in a population, so they don't have to be things that you can control, they just have to be factors that would be of direct interest and someone else could repeat your study on similar or the same age groups, habitat, season, and so on. And a feature of fixed effects is that anything that you learn from the experiment regarding those particular treatments apply to those particular treatments, and that's it. You can't really extend to treatments that were not included in the study. So maybe a typical kind of example in ecology would be an experiment that Chris Harley did a number of years ago. It's a field transplant experiment in which he took copper rings to exclude herbivores and looked at the growth of red alga. He did this in the low intertidal zone and in the mid intertidal zone, a total of 64 plots in a completely randomized design. And so these are both, this is a factorial experiment, all combinations of the two levels, herbivores presence and absent, and intertidal zone low and mid. All combinations were conducted and replicated, and they could be repeated. Someone else in Chile or somewhere in Europe could repeat the experiment. And he observed, in this case, that algal growth there was an effect of herbivore treatment on algal growth, but it depended on which region of the intertidal the experiment was carried out upon. Once again, here's a description of the linear model that would be used to analyze it. Again, x1 and x2 are the two factor levels, and the parameters that describe them will look like that. And if you were going to analyze these, you would analyze them using LM, because that's what LM is for. It's linear models for fixed effects. And the two variables here are herbivores and zone, referring to zone in the intertidal. And the ANOVA table would look like this. Chris gave me his data, so I was able to analyze it. I could produce the graph, and I could show you what the ANOVA table looks like. And the way you probably learned ANOVA analysis of variance generally is that the F ratio is calculated by a ratio of mean squares. The mean square for the effective interest divided by the residual. And that makes great sense because the residual is the only source of random variation in the experiment. And the mean squared for zone is a combination of the effective zone plus sampling error. And the ratio cancels out the sampling error and allows you to detect whether there's a an effect of the zone over and above what you would expect from sampling error. And for this case, that was not true of zone, but it was true for herbivory, and it was certainly true for the interaction between the two. So I don't know if you learned ANOVA tables and dividing and calculating F ratios yourself, but you probably did, and you would have divided the mean squared from the particular source by their residual. Random effects are different. So random effects are categories of a variable that are actually randomly sampled. Or at least we pretend they're randomly sampled when we analyze the data because they're sort of arbitrarily sampled. And these factor levels represent groups or clusters of measurements or units that are sampled together. And the difference between a random effect and a fixed effect is that an experiment with the same treatment levels really couldn't be repeated by someone else. So if you were going to carry out an experiment on different individuals belonging to families, say that you created in the lab, those families are made up of siblings, and the siblings are all going to be measured, but they're grouped by the families in which they occurred. And family is therefore, in the spirit of analysis follows design, family would need to be included in the model. But it's a random effect. You could never really repeat those families again in a repeat of the experiment itself. So randomly sampled subjects that are measured repeatedly, randomly placed transects of multiple quadrats in a sampling survey, arbitrary field plots, growing plants for some agricultural experiments, environment chambers in which you may have carried out, in which you may have aquariums or trays of plants. And the idea behind random effects, and certainly this is the way that they'll be modeled, is that the measurements come in groups. Siblings come in families. Repeated measurements made on the same subject are grouped by subject, and so on. Quadrats are grouped by the transects in which they were placed, and so on. Those groups are treated as though they were randomly sampled from a population of groups that you might have sampled but didn't. And one difference in this case then is that because the groups are randomly sampled from a population, any result obtained on the randomly sampled groups can be applied to that entire population. Generally, usually, the random effects are actually of no interest whatsoever. You don't really care about this particular plant. It's not a gripping question what the effect of this particular plot in this particular field is. And it's not particularly interesting the attributes of the environment chamber or environment chambers that you happen to use to conduct your experiment. Those effects really are not very interesting to you. But it is the case that often the variance associated with those random effects, the differences between the chambers, the effects of the individual field plot, they are of interest in some ways. One, you have to incorporate them in your design, but in some cases you're actually interested in the variance components because you want to do something like estimate heritability, contribution of genes to variation in measurements between families, or you want to estimate repeatability, the extent to which you get the same measurement over and over again if you repeatedly measure the same subject, and so on. So but a key feature of an experiment or an observational study in which there are random effects is that the units of sampling within that are grouped by those random effects, the siblings that are grouped by family, the repeat measurements that are grouped by subject, failure to incorporate them in your analysis would commit pseudoreplication. Siblings belonging to the same family are not independent because they share genes and environment in common. And so we can't pretend in an experiment that they are randomly sampled from some population. However, if the families themselves are randomly sampled from the population, and we incorporate family as a random effect in the model, you're free. You avoid pseudoreplication. And so modeling random effects will prevent you from committing pseudo-replication in experiments. Here's an example. So here's a factorial experiment, again involving two treatments, but now one of the factors is random. So this is a study by Fatuma and Philippi on a moth called the fall cankerworm. So this is a caterpillar of some agricultural importance, and the species in the caterpillar phase feeds on the leaves of hardwood trees in eastern North America. The adult female moths are wingless. Many reproduce clonally. I didn't know that. They produce only daughters genetically identical to themselves. Insects are amazing. And the question that was of interest to them was, first of all, what's the effect of different tree species on the growth of this cankerworm? But also, do the clones vary in their growth? In other words, is there a genetic component to growth on different trees? And the question itself is about the trees. That's the fixed effect of interest. But the moths are sampled by clones. And so the experiment, in this case, was carried out using multiple individuals from multiple clones, but only the clones were randomly sampled. So the design of the experiment was 9 female moths were sampled from a population in New York. Let's say they were randomly sampled, and then the larvae were raised from those 9 clones on leaves of 4 different tree species, and then after 15 days they measured growth. So there's the log of growth along the x-axis of the 4 different tree species that they used, and this is what's called an interaction plot. We're looking at the mean of a particular clonal group on those 4 different tree species. So every clone was tested on every species. And just by looking at the graph, we can see that there is some variation in the growth rates of different clones on those 4 species. And we can also see from the plots that the lines aren't necessarily parallel. In other words, it's possible that some clones are, say, better on one kind of tree relative to their performance on a second species of tree compared to other clones. And that specific factor was one of the things of greatest interest and motivated this experiment in the first place. So here's an experiment that's carried out, again with 2 factors, tree species and clone. And what's special about clone is that it's not a fixed effect, it's a random effect. And a random effect introduces another level of randomness, another source of variance to the model, and then must be incorporated in the analysis. So caterpillars from the same clone are not independent. We can't just analyze this as though there were hundreds and hundreds of independent observations. We have to somehow take into account the fact that the caterpillars are grouped by clones. And that to some degree, looking at the effect of one tree species on another is going to be determined not so much necessarily by the number of moths that grew better on it, but by the number of clones that grew better on it compared to other tree species. So analyzing these data with LM would be wrong, because LM assumes that all of the factors you're incorporating are fixed. To do so would commit pseudo-replication. And so somehow the variance among the clones has to be taken into account in order to properly analyze these data. And my general rule for everybody when I'm reviewing papers, when I'm reviewing theses, when I'm writing papers, and when you write papers, is that when you carry out analyses like this, always report the degrees of freedom. If you're going to provide an F statistic in your text, also provide the degrees of freedom, so we can tell. Because we can tell from the degrees of freedom that you provide whether you've assumed that caterpillars are independent or not. It will affect the degrees of freedom. So always record them. Okay, so clones, randomly sampled. It's another random factor, another source of random variation. If you were to repeat this experiment, you wouldn't use the same clones. They don't even exist anymore. You'd go back to this population, and you would get different clones, and as a consequence, the results that you would get would be would differ. But in this case, not because of variation among caterpillars individually within clones, but the clones themselves. So there's now 2 layers of sampling variation. And that must be taken into account. So, as I mentioned earlier, and it is true in this case, unlike the fixed group, the means of the specific random groups, clones, are not of interest. It's like the chambers, the field plot, and so on, or the families. They're not so interesting, but the variance among those random groups might be interesting, and in this case it would be used to estimate heritability. I won't go into that other than just say that variance components can be interesting in their own right, but the specific clonal values are not. Another feature of random effects is that it makes it harder to analyze if the experiment should become unbalanced through, say, differential mortality. And in the old days, we used to calculate these things with computer packages that analyze the data like LM. You would get your ANOVA table, and then you would have to redo the F ratios to calculate them properly, to take into account the fact that you had random effects, and there were tables in Sokal and Rolf that explained how you actually calculate these F ratios when there are random effects. But if your design became unbalanced, sorry, those formulas don't work. And so this is the ability to actually specifically model a random effect using packages like R represents a substantial advance that happened in my own life. I can remember a fellow graduate student when I was a graduate student who'd done this experiment, and things went wrong almost from the beginning, and they ended up with a severely unbalanced design, and they went to the statistics department to analyze it. They wanted to calculate F ratios properly, and they said, sorry, you really can't do it. The only way you could ever really analyze your data is if you threw data out of those cells that had basically you balanced your design by randomly throwing away data until the sample sizes were the same in all combinations. We don't have to do that anymore. We have methods now based on likelihood that will analyze unbalanced designs, but they are actually conservative. They remain approximations. Okay. How do you know when you have random effects in your study? You have random effects whenever your sampling design is nested. Quadratic within transects, transects within woodlots, woodlots within districts. Whenever your measurements are clustered spatially or temporally within randomly sampled units, like blocks. Blocking is a feature of experimental designs that we talked about previously, and blocks are analyzed as random effects. Whenever you divide up plots or families, clones, pawns, and apply separate fixed treatments to subplots of those plots. When you divide families in two, and one group of siblings gets one treatment and another the others. Whenever you make measurements on related individuals, and whenever you measure subjects or other sampling units repeatedly, that's when you have random effects. So how do you analyze random effects? You use linear mixed effects models. So I'll explain a little bit what they look like. Most stats packages will analyze data using linear mixed effects models. We used to use JMP in the undergraduate course here, and in that program, the package assumed that all the effects were fixed unless you sort of burrowed in and checked some little box indicating that one of those factors was random. So in that case, designing or designating a factor as random took extra work. You had to read the manual. In R, as I said before, LM is for fixed effects, but there are packages in R for analyzing data that includes random effects. So what we'll be using in the workshop this week is the LMER function, which is part of the LME4/LMER test package, or LME in the NLME package. So both of those are available and both of them are used. I used to use this in the workshops until I discovered that somebody did a simulation to show that the methods for calculating degrees of freedom are more accurate in the LME4 package. So that's what we're going to use. So what are linear mixed effects models? They're linear models that include both fixed and random effects. They model different error variants for every level of random variation. Estimation and testing are based on restricted maximum likelihood, and it's able to take advantage of, it's able to handle unequal sample sizes, though the methods are approximate. P-values are conservative in this case when designs are unbalanced, and I already told you which package they're implemented in, and that's what we're going to try. I'm going to give you show you an example of the simplest possible mixed effects model, and it's a study of repeatability. The question is basically, if I measure this thing, these insects, multiple times, do I get the same measurement each time? So Patrick Nossel did this. These are Temima christinae, walking stick insects, a wingless herbivore. And so he has been conducting field studies in California on populations of this insect. And so he collected individuals and placed them each in envelopes. And then had all these envelopes in a box. And so he took each envelope, set each insect in the envelope on a platform, took a photograph of it, and then one of the measurements that he made was femur length, shown here. Then he put the insect back in the envelope, back in the box, did the next insect. And then once he'd gone through all of his measurements, he started all over again. Went back to the specimens that he had already measured, took them out of the envelope, repositioned them, put the camera overhead, took a picture of them again, and then digitally measured femur length again. So he didn't just take 2 photographs of the same mount, essentially, and then measure them twice, or didn't measure this length twice in the same photograph. No, he went, actually, put them back, brought them out again. And what he observed was that he rarely got the same measurement twice. So these are small things, and measurements are made with error. And it's not that it's a mouse error as to where you actually click here and there. It's what's the angle of the camera relative to the position of the animal, and what is the position of its leg? Did you actually see all of it, or was maybe some of it obscured by the body, and so on. So what that means is that measurements are made with error. Get used to it. But it's possible, if you take repeat measurements, to estimate the measurement error. Or in contrast, the repeatability, what fraction of the variability among measurements is actually real. And how much is measurement error. So this is sort of a dot plot, and I've just arrayed the individual walking sticks, 25 of them, along the x-axis, and this line just connects the 2 measurements made of femur length on the same individuals. So a demonstration that it's rarely the case that you get the same measurements twice. But how bad is it? So here's the design. The design is that there are 25 insects and 2 measurements are made within the same insect. So it's a repeated measures design. You can think of it as the 2 measurements are nested within individual insects. And we're going to fit a model that is a linear model. It looks like linear models that we've seen before. It's going to have a constant, but it's now going to have 2 sources of random variation. And one is the variation among the individual insects that you randomly sampled, and the other is the error, which in this case is all measurement error. So the individual bugs in this case are the random groups. We call them groups because we have repeated measurements made of them. So again, model has 2 parts. That's the best way to think about it, and that's how you'll sort of be doing it in R, is to say, okay, we're going to separate the fixed effects from the random effects when we actually construct the model, even though they'll be put together. The only fixed effect in this case is just the grand mean. It's a constant, an intercept that is called a fixed effect in this case. And then the random part is the measurements of individual bugs. But the second random part is the random sample of bugs you have in the first place. So here's, if you use the LMER test package, the construction of the linear model in R. So the LMER, the response variable is femur length. We model it as a constant, and that's the fixed part. And then the random part is modeled with parentheses and a vertical bar. And what this construction means is that we're fitting now at the same time as we have an overall grand mean, which is fixed, we're going to have random intercepts. Every individual is going to have its own intercept, which is its true bug measurement. And individual tells R what the random groups are in this case.
So, if you want to add multiple random effects, which is like plus and then in brackets,
Correct.
blood, 1, and then plus.
Yeah,
plus,
each random effect would be in its own brackets. Yeah, so the formula itself has a fixed part and a random part. Other than that, it's just like any old linear model. So the fixed part of the formula tells R to fit a constant to the data, estimate the grand mean, based on the fitted values of the random groups, the individual bugs. And then the random part of the formula instructs R to fit a constant, an intercept, to the two measurements within each individual. And so this yields a fitted value for every individual walking stick. Now we're not actually interested so much in the individual walking stick, but we are interested in the variability among the fitted values. And if you ask R for the fitted values for each of the bugs, it'll give them to you. Just like using fitted will provide you with the fitted values in a linear regression using LAM. So what I've done here is I've shown you again this graph of the individual walking sticks and the femur lengths, and the two dots are once again the two measurements. So this is the same graph that I've shown you before, but I've removed the line. And instead what I've done is I've shown you what the fitted values are for each of the individuals from the model. Now you might think that the fitted value for every individual is simply the average of the two measurements. But that would include variation among individuals and some measurement error itself. And so R basically assumes that there's a regression to the mean effect built into these repeat measurements. Namely that it is likely that the most extreme measurements, even if there's two of them, will be overestimating the true value for that bug. So those that are extremely small and those that are extremely large will tend to be overestimated. And then if we were to measure them again, we would get closer to the true value the third time or the fourth time. So these fitted values are not just the averages within each bug. They're called LUPs, best linear unbiased predictors. So they take into account this regression to the mean. And you'll see them referred to as shrunk. They have less variation among these red lines than variation among the averages of the twin measurements per insect. Once we fitted our random effects model, we can extract the variance components. And as I mentioned before, there are two levels of random variation in this design. There's the residual from the model, and in this case, that's the variation within random groups, because we fit an overall constant and we fit separate intercepts to every individual. And the residual from that model then is an estimate of the variance among the measurements within bugs, among the repeat measurements. But there's a second variance component that's estimated, and that second one is the variance among the bugs, in the true measurements among the bugs, the variance among the random groups. And these are the two quantities we need to calculate the repeatability. The repeatability is the fraction of the variation that's real, the fraction of the variation that's among measurements. And so it's calculated as the estimated variance component among individuals, which is this here. So this is standard deviation, you have to square it to get this one. Squared. Over the total variance, which is the sum of those terms. And in this case here we get a value of 0.75. So 75% of the variation among all these measurements is real. It's variation among bugs. But 25% of the variation in the measurements is measurement error, which is also reasonably high. It's better to know that than not to know it. I'm going to give you some other examples of experiments that contain random effects, just to sort of get you used to the idea that what random effects look like and how they're modeled. So this is a repeated measures design as well. Again, the random groups are individuals, and each of them is measured multiple times. I'm showing data for 4 times. So here's an old experiment that looked at sensitivity of the goldfish to colors to light of different wavelengths. You all probably know what a goldfish looks like. This is what goldfish used to look like before artificial selection made them brightly colored. They're kind of dingy, orangey brown. So what the experiment does is, does anyone know what an optomotor experiment is? An optomotor response? You heard of it? You all have this sort of innate tendency to sort of follow objects that are moving. If your eye does this. If you're a passenger in a car and you're driving along the highway, your eye sort of leaps from telephone pole to telephone pole. If you put a fish in a circular chamber that has vertical stripes on the wall, and then you turn this chamber very slowly, the fish will attempt to remain next to one of the stripes. It'll do this completely autonomously, and it never habituates. And you can basically dim the light. You can provide light of a very specific wavelength and make it dimmer and dimmer and dimmer. When the fish stops following the line, it means it can no longer see it. So what this experiment did is it subjected the same fish to a series of wavelengths provided in random order. And light sensitivity is a measurement essentially of how well able they are to see very low levels of illumination. So high light sensitivity means they see orange better than any other wavelength. And it's an interaction plot, so this joins the measurements made on the same individual. Each individual was tested just once. So it's a repeated measures design, but each repeat measure is on a different treatment, a different light treatment. And what we're interested in knowing is whether there are differences among wavelengths in there and the sensitivity. And that's our fixed effect of interest. The random effect that we must take into account is the fish. Why do we have to include it in our model? Well, we measured the fish repeatedly. So it's in there. Repeated measurements made on the same individual are not independent. But if you include fish in the model, then you've avoided pseudo-replication. some of those goldfish data in the workshop this week. Here's an experiment that we did in my lab, which has a design called a split plot design. So a split plot design is a two-treatment experiment in which you have whole plots that receive one treatment or another. And then within each of the plot, it's divided and different levels of another factor are applied to each side. So in this case, it was a split pond experiment, really. So each of 7 ponds was divided, and 2 competition treatments were applied, one randomly chosen. One side received this species as the competitor, the other side received this species as the competitor. The target was added to both sides. And then whole treatments were supplied to the ponds were predators. So some ponds had predators added, and some ponds did not have predators added. So this is an experiment conducted on stickleback, in case you hadn't figured that out. And the predator in this case is cutthroat trout. So split plot design. I'm interested in the effect of predation. I'm interested in the effect of competition, but we have repeated measures here. We have repeated measures in the sense that we have each pond is measured repeatedly. We have a result from this side, we have a result from this side. And we have all the individual fish that were collected from each pond, which are nested within each side. So the predation treatment is fixed, we're interested in that. The competition treatment is fixed, but the pond is random. So, other than that, it's just like any other linear model. But it makes assumptions which are like those of LM, but a little bit different as well. So, the usual assumption when we're carrying out analysis of variance, linear regression, and so on, is that the errors, the residuals, are normally distributed. So that's familiar. And the second part of that is that not only are they normally distributed, approximately, but the variances are homogeneous. So the residuals have the same variance in every group, regardless of the values of the fixed effects. A second assumption is that, in this case, when there are random effects, that the groups are actually randomly sampled from some population of groups. So in the case of moths, moth clones selected from a moth population in New York, or ponds sampled from a universe of ponds that might have been. We had only 7, but we model them as though they're somehow sampled from a population of ponds. And finally, we assume that every pond, every level of the random effect is sampled from a population that itself has a normal distribution. So the moth effect, the individual moth effects, clonal effects, or the individual pond effects, if we could estimate those pond effects, we would see that they had a normal distribution. So the analysis assumes that as well. You assume that replicates within the groups are randomly sampled, so the siblings are randomly sampled from the families, and that they're sampled without bias. Also that there's no carryover between repeated measurements on the same subject. So in this experiment here, one way to ensure that there's no carryover is to avoid starting at low wavelengths and gradually all going in the same order, increasing wavelength, increasing wavelength, increasing wavelengths, but rather to provide the treatments in a random order, so that there's no correlation between consecutive measurements. It turns out in this case the optimum motor response is so fixed that it doesn't matter, but in many other kinds of experiments there's the possibility that what you've just measured will influence what you next measure, even if it's in a different treatment. And in a situation like this, you have to make sure that when what's going on one side of the pond doesn't affect what's going on the other side. Finally, sphericity is an assumption of linear models in general. And I mentioned this once before in the experimental design lecture that I gave earlier. And the sphericity assumption is that the variance of the differences between pairs of factor levels are all equal. And I already showed this example as a case in which that assumption might be broken. And that assumption is likely to be broken when one of the fixed effects, in this case time, is measured consecutively rather than in random order. So time always must be measured consecutively. And that's one of the reasons why time as a factor is often very problematic. And I mentioned also that there are methods to deal with this. In this experiment, there were 4 enclosures. In 2 of the enclosures, rodents were removed, and the other 2 enclosures they were not. The response variable is the number of colonies of ants. This experiment was carried out in the desert in the American Southwest. And so rodent treatment is a fixed factor, and the temptation is to include also date. Plot is a random effect, and it must be a random effect because when it's measured repeatedly, well, we have to repeat measurements made on the same plot over and over and over again. And I just mentioned this again because date might cause you to violate the assumption of sphericity. But here's how you would model it. Number of colonies. The formula has 2 parts, the fixed part, treatment and date, and then the random part. You're modeling a separate intercept for every plot, the plot effect. So, including census date as a factor likely leads to violation of the sphericity assumption. And what that means is that measurements made on the same plot closely adjacent to one another, the difference between measurements made closer together inside is likely to have a different variance than the difference between plots measured at more distant points in time apart. And that is what the sphericity assumption means, what it means to violate. And it increases type 1 errors, in my experience, by an order of magnitude, but there are ways of correcting for this. R, of course. So there are tests for sphericity, and there are corrections for it. So I'm just telling you this because be careful when time is a factor in your analyses. It's more perilous than you might imagine. Where do you go for advice on stuff like this? Suddenly the kinds of experiments that we're analyzing and that I'm showing you examples of are getting more complicated. The fixed effects, the random effects, there might be more than one fixed effect, there might be interactions. Where do you go for advice? What's my source? For analyzing data that includes both fixed and random effects. So I brought this book in the first lecture in the course. I really like this Quinn and Keough book. I find that it's very well written, it's very clear, it identifies the problem. It won't tell you anything about R. There are a variety of books out there now on analyzing mixed effects models in R. So I have links to them on the books tab at the course. The original one, the classic, is Pinheiro and Bates mixed effect models in S and S+. And that uses, it's all about the package NLME, not the one we're going to use this week, but the first chapter is highly readable and will give you a good idea of what it means and how you can model random effects, give you some more intuition. Zur has a book, Mixed Effects Models and Extensions in Ecology with R. I've looked at it a couple of times. I like the first chapter of this one for a start. I find this one more complicated to begin. Also, there's a paper which tells you how to, which sort of gives an overview of the LME4 package, which is the one that we'll be using. But at the moment, there's no real easy guide to fitting mixed effects models in R. Well, there's a variety of sources, and and the World Wide Web. There's lots of discussion on the World Wide Web about how to analyze specific designs, how to analyze specific designs as well. The discussion paper for next week. Do we need mixed effects models at all? So Murtagh has a sort of a curmudgeonly attitude towards this very question. And I have 2 presenters signed up. And I'm looking for 2 moderators. So if you're interested in moderating this particular discussion, come up at the end of the lecture, which is now, to volunteer yourself. Otherwise, R will do its thing and randomly sample names, and I'll be in touch by the workshop on Thursday, if not before. And I think that's it. Yes, it is. Okay, thanks. Any questions?
I ever moderate, I hear pennies. That's like,
You can keep them if you want.
that's okay.
They're gonna be worth something someday.
I have to go pick up a